In this video, we will discuss about effect of microglial depletion timing and the chemokine receptors on cognitive behavior. So far, we have discussed that when we inhibit this CSF1R receptors, then it will leads to the depletion of these microglials and also the revival of the oligodendrocytes and oligodendroglials that we have discussed in our previous video. So in this video, the researchers want to check out or to find out that whether there is a effect of this microglial depletion timing, and whether if this microglials after depletion of these microglials for a certain period of time and they are revived, whether it will again have their impact in terms of this decrease of these oligodendrocytes and decrease in this novel object recognition test or not. So we will discuss those things and their results in this figure or in this video. So panel A describes the plan of the experiment where the xenograft is implanted, and then after 3 days this CD19 CAR T cell treatment is done, and then the normal depletions of the CSF1R is done using on day 14 to 28 after the CD19 CAR T cell treatment, and which they call it as late CSF1 inhibitions. And from 28 to 35 days they again stopped the CSF1 inhibitions, and that will cause the repopulation of these microglials. And there is another condition that they have used where they started CSF1 inhibitions from the day 0, that is the time they induce CD19 CAR T cell treatment, and from this day 14 and so this is the experimental plans that they used. The results are in the panel B, where we can see that for this mock T cell control, we can see a decrease in the microglial depletions, and for the actual experiments we can see that for the late CSF1 conditions we can see a decrease in those microglial populations, and when the microglials is repopulated in the case of early CSF1 we can see the repopulations of these microglials, and also for this late CSF1 conditions repopulations we also see a regrowth of these microglials. And thereafter, when they check this reactive white matter microglia, they can see a decrease in this by reactive white matter microglia for the early CSF1 conditions. But for the case of this late CSF1, they do not see a for this late CSF1 conditions, they do not see a too much decrease in this reactive white matter microglia populations compared to the vehicle. But for this early CSF1 condition, they see a decrease in this reactive white matter microglia conditions. And then they also check this white matter oligodendrocytes, what its impact, repopulation of this microglia impacts on this white matter oligodendrocytes, and they can see for the early CSF1 inhibitor plus repopulation conditions, they see a decrease in the oligodendrocytes, but not decrease. They do not see an increase in the oligodendrocytes. But for the case of this white matter oligodendrocytes conditions, they see a significant increase in these oligodendrocytes conditions compared to the vehicle. And similar results they have seen for novel object recognition test or NORT, where they have seen that for the late CSF1 conditions they see a significant increase in this NORT recognition ratio compared to the early repopulations, and both of them are results are compared with the vehicle. So for the early CSF1 inhibitions plus repopulation, they do not see a significant increase, but for late CSF1 inhibitions plus repopulations see a significant increase in this NORT test. Thereafter, they decided to check another approach, whether chemokines also has a effect of this microglial depletions or not. So here they do two things. First, they use this one antibodies, and second one, they inhibit this CCR3 using they neutralize CCR3 receptors using CCR3 antagonist, and in another case they use this CCL1 neutralizing antibodies to neutralize the CCL1 chemokines, which are found to be upregulated in this CAR-T9 therapies. And the plan is that after first the xenograft is implanted, then after 3 days they use CD19 CAR T cell treatment, and from day 7 to day 28 they induce either the CCL1 neutralizing antibodies or CCR3 neutralizing antibodies, or the control this PBS. The results what they have found out is that for the case of CCL1, they do not see a decrease in this total white matter microglia, but for the case of CCR3 inhibitions, they see a decrease in the white matter macroglia. And when they check the reactive white matter macroglia, they can see that reactive white matter macroglia is reduced for the CCR3, but there is no change when they use CCL1. And when they check the oligodendrocytes, here also they are able to see that the increase in these white matter oligodendrocytes when they use CCR3 inhibitions. And for the NORT test also they see an increase in this recognition ratio of the NORT test for the CCR3 inhibitions compared to the CCL1 inhibitions in both the cases, for oligodendrocytes, white matter oligodendrocytes, and also for the NORT cells. So all this indicates that it's the receptors inhibitions that cause the revival of this cognitive behavior, not this CCL1 chemokine inhibitions. So the timing also has an effect, that the late inhibitions will cause improvement or will improve the cognitive behavior, and the receptors with CCR3 inhibitions will also improve the cognitive behavior. And in all these conditions they also checked whether the tumor clearance is occurring, and they do not see any impact of the tumor clearance of these reagents that they used to inhibit or revive these white matter oligodendrocytes. So that's all for this video, and this is the last main figure that I presented in this paper. If you need to understand more about this topic, or if you have any questions, you can go to the reference that has been provided in the description, and you can also mention in the comment so that I can read and try to resolve those doubts in any of the videos or by replying to the comments directly. Thank you for your time.